ResistSense begins with an assembled Escherichia coli FASTA file. I select a genome to start assessing resistance across 5 antibiotics. While the assembly is being interrogated, ResistSense verifies genome quality, required annotations, and molecular target context before running endpoint models. A classifier can sound certain when a sample is incomplete, unfamiliar, or poorly supported. Calibrated probabilities are challenged by conformal uncertainty, model agreement, and genetic out-of-distribution distance. Missing model, annotation, target, calibration, or quality evidence produces a machine-readable no-call. Marker absence is never proof of susceptibility. Evidence is separated and tested before release. The prototype remains research use only, and every endpoint requires standard laboratory susceptibility testing. The assessment presents 3 outcomes: a resistance-associated genomic signal, a susceptibility-compatible signal, or a no-call when evidence is insufficient or contradictory. Ampicillin displays resistance-associated evidence, and the visualization shows its cellular envelope remaining intact. Ciprofloxacin and cefotaxime display susceptibility-compatible signals through mechanism-informed views. Gentamicin illustrates ribosome stress, mistranslation, progressive membrane leakage, and a time-compressed cellular endpoint. These animations are educational illustrations, not microscopy, viability measurements, or treatment simulations. The trimethoprim sulfamethoxazole endpoint remains a no-call, so the cell stays neutral and no biological response is inferred. Firewall replay exposes the immutable decision path. Statistical association, known biological markers, target evidence, and safety checks remain separate. The interface shows calibrated probability, conformal set, marker evidence, target status, distribution shift score, and the guarded output. A confident prediction can still be blocked when the sample is out of distribution. The scientific outcome is locked before any optional language model audit. GPT-5.6 Sol performs a constrained evidence conflict audit. It receives only allow-listed structured evidence, never raw FASTA, file names, genome checksums, or personal data. A deterministic validator rejects altered statuses, probabilities, identifiers, counts, or required warnings. The model can audit and communicate evidence, but cannot modify the scientific result. The executable adversarial suite tests decision integrity, evidence integrity, privacy, clinical language, and availability. The evidence passport records provenance, PIN tools, policy digest, model coverage, and the OpenAI boundary. The frozen evaluation contains 2,909 curated E. coli assemblies. Grouped validation keeps related genomes inside one partition, while prediction autopsy exposes blocked and residual held-out errors. 6 safety barriers make abstention a visible feature, instead of hiding uncertainty behind one accuracy number. Finally, the exported JSON preserves antibiotic-specific statuses, calibrated probabilities, evidence tiers, safety reasons, and the laboratory confirmation requirement. ResistSense does not prescribe an antibiotic. It makes genomic evidence traceable, challenges unsafe confidence, and abstains when evidence is insufficient. Predict, challenge, abstain.